Always good. We have two speakers today, Marina Sinota from UCSF and Mildred Cho from Stanford. And I believe Marina is first, so come on up, Marina. And please join me in welcoming.
All right. Hi, everyone. I hope you can hear me. My name is Marina Sinota, and I'm a professor at UCSF and also the interim director of Baker Computational Health Sciences at UCSF. And today I'm going to tell you a little bit about my work. But before I start, as I'm saying all this, I'd like to dedicate this talk to Dr. Atul. Many of you guys knew Atul. He was an incredible person, a visionary scientist, a mentor. He was a devoted husband and father, and a very dear friend to many of us. He was my mentor. His career was defined by his unwavering belief in the power of data to drive discovery, equity, and better health outcomes for all. He was an incredible researcher and an incredible person. I put down a few bullet points. We all love data and statistics, so I thought I would tell you a little bit about him if you didn't know. He got his undergraduate medical degree from Brown University. He did residency, fellowship, and PhD at MIT and Harvard, and then he spent a decade at Stanford as a professor and also the chief of the Division of Systems Medicine, and he was on faculty at UCSF for the past 10 years. He was the inaugural director of the Baker Computational Health Sciences Institute, and he had a role as the chief data scientist for the state of California. He authored many, many papers, trained a large number, over 100 undergraduate, graduate students and postdocs, and he received many awards. I'll just mention a few. The Morris Collin Award of Excellence. He was part of the National Academy of Medicine, American Academy of Arts and Sciences. He was named as Open Science Champion of Change by the Obama administration. And all of these honors and accomplishments are amazing, but truly what made him special is the kind of person that he was. I'll talk a little bit about his scientific career first. He was one of a few scientists that I know who's redefined his career several times. He started out his research career with Zach Kohane, and you actually can see, I think one of his most cited papers was a PSB paper on relevance networks over here that he co-authored with Zach. And really, it was very early on looking at co-expression networks using microarrays, and obviously this work has made a tremendous impact. It was hard for me to find pictures of that face because that's before my time, but I did find a few. I think the one on the right is from a newspaper article about the work. Then he came to Stanford, where he defined this field of translational bioinformatics. And that's around the time when I met Atul as a graduate student. This was part of the AMIA proceedings. And then he's did many different, of course, many different studies, but I'll just highlight a few. One was a collaborative effort. I wasn't part of this, but in collaboration with Russ, Ewan Ashley, Mike Snyder, and many others. This is around genome interpretation. And one thing that he believed very strongly is that we shouldn't just write papers. We should really go into translation and move these projects to help patients. And this resulted in starting of a company called Personalis. The work that I was leading in his lab as a graduate student, and I'll come back to it when I talk about some of the work that we're doing now, is around using gene expression for drug repurposing and drug discovery. This was published in Science Translational Medicine. It was part of my thesis and also led to starting of a company called NewMedi that was led by Ginny Deshpande. And these are some photos from that early era. I don't know, Russ, what you and Atul are talking about, but you seem pretty engaged there. This one at the top is the first lab gathering. This is the original Butte lab. We might have carved Atul's face on a pumpkin in a Halloween pumpkin competition, and then, of course, had many, many happy moments then. So he spent 10 years at Stanford figuring out how to use different types of data with a focus on omics data for both therapeutics and diagnostic discovery. And then he moved to UCSF and reinvented his career a third time, focusing on clinical data sharing. This is an opinion piece that came out only a few years ago, talking about AI from starting pilots to scalable privilege. And he made a huge impact in that space as well. And these are some photos from our UCSF era, and I was fortunate to join him at UCSF. I think that picture on the left top is from a Twitter post, and I don't know how many times it was reshared. It's like thousands, if not more. The one on the right is our first Institute research retreat. You can see a tool in a hat. This is our building being built, and we went to visit the construction site. We all had to wear hard hats for that. And then this one on the bottom right is a reunion that we had about a year and a half ago that I hosted. I don't know if many of you know, but Atul was an avid Star Trek fan. The picture on the left is one I photoshopped as a graduate student for my PhD thesis defense. But AI has gotten much better. So the one on the right is generated by AI. It's much smoother than what I could do before. So the technologies are improving, not only in the discoveries that we make, but in image analysis too. I wanted to share some of Atul's quotes and wisdoms with you. He truly believed in the power of data and data sharing. Hiding within those mounds of data is knowledge that could change the life of a patient or change the world. He really believed not into developing methods for methods' sake, but really asking impactful scientific questions. Data, unlike oil, multiplies when shared. Once we let the data flow, lifelong discoveries become possible. If you want to change the world, you can't just keep writing papers about it. The science continues in the startup companies, and he truly did that throughout his career. Finally, last one is one of my favorites, is credit is infinitely divisible. Give it away every chance you get. There's always plenty left for you. So I met Atul when I was interviewing for the BMI PhD program, and he was giving a job talk at the retreat in Asilomar. I was blown away by his charisma, by his excitement, and I really liked what he was. Prior to that, I was doing research in comparative genomics, which is very cool, but I was truly amazed that we could use the same skills, informatics, to help patients with disease. And that was the whole field of translational bioinformatics that he was pitching at the time. So, in the beginning of the school year, this was September 2006, I wrote him an email asking for a rotation in his lab. I liked it so much, I did 2 quarters of rotations in his lab. And then about a year later, I've asked to join his group. And I feel incredibly fortunate that I got a chance to work with him, both as a graduate student and then later in my career. He's had a tremendous impact on my career and my life. After my PhD, I spent a few years at Pfizer, and he was instrumental in bringing me back to academia, first to Stanford for a year, and then I started my lab at UCSF in 2015. Nearly everything I know about doing science, I learned from Atul. He taught me how to ask questions, how to think big, how to mentor and bring collaborative teams together. He taught me the importance of communication and telling a story. He really elevated people around him, making them feel good about themselves and the work that they do, which in turn inspired them to grow. He was incredibly loyal and dedicated to his work and his family, and his energy was contagious. His loss is deeply felt, but his influence endures in the lives he touched, the data he unlocked, and the futures he helped shape. So with that, I'd like to tell you a little bit about the type of work that I do in my lab and frame it in a way that demonstrates Atul's influence on my career. So I'll tell you 2 stories today in the diagnostic and therapeutic space. And in my group, we use data, all different types of data, to better understand disease across the lifespan. I do a lot of work in reproductive health, pregnancy outcomes like preterm birth and recurrent pregnancy loss. We study autoimmune disease. This is stemming from my graduate school interests. And then we have a few projects in Alzheimer's disease and neurodegeneration, and that's what I'm going to focus on today. As part of the Baker Institute, and I think one of the things that drew Atul to UCSF and the UC system is the possibility of clinical data. So what he's built over the past 10 years is an infrastructure called Information Commons, which contains de-identified clinical records from all the patients at UCSF. So it's about 8 million patients, hundreds of thousands of encounters across the past 40 years. We have clinical notes, we have imaging data, and then we have some limited genomics. All the data is de-identified, accessible, and computable. We also have access to data across the UC system. So this is 5 UC campuses, well, 6, $12 million of data, 9 million patients, some cancer genomes, and you can really think of this as a built-in discovery and validation system. We have deep data at UCSF and broad data across the UC system. And it's an amazing opportunity for development and application of AI and machine learning approaches. So I've had a very talented MD PhD student, her name is Alice Tang, who was interested in exploring this data in the context of Alzheimer's disease. So we started by identifying a cohort using ICD codes as a first pass, and we wanted to see what is it that makes these Alzheimer's disease patients special. So here is data from UCSF, each dot is a patient, and the dimensionality reduction is done on the clinical profiles. We're very used to seeing these types of plots for molecular data, but here this is based on their diagnosis codes excluding Alzheimer's disease. The controls are shown in blue and the Alzheimer's patients are shown in red. And you can see there's no clear separation, but there's a bit of a shift. We're able to replicate this at Mount Sinai. This is early work. We did not have the UC-wide data at this time, so that's why we went to collaborators at Mount Sinai for validation. We can build comorbidity networks. We can start to understand what are other diseases that are associated with Alzheimer's disease. And you can see that in Alzheimer's, the network is a lot more dense than in the control population. Not too surprising. We can also look at sex differences. We know that Alzheimer's is a lot more common in women than men. Why is that? What is it that makes women different from men when it comes to neurodegeneration and Alzheimer's? And you can see the differences in these types of networks. We can look at association studies. Not surprisingly, some of the most associated comorbidities are MCI and dementia. But as we go down the list, we can see things like urinary tract infection, hypertension, hyperlipidemia. And we see some associations that are inversely associated with Alzheimer's, like cancer. You can see that there's pretty good concordance across data at UCSF and Mount Sinai, even though these are completely different patient populations. This is looking at sex-specific differences. So there are some associations that are common to both males and females. However, we see quite a few that have a greater effect size in females, including hypertension, osteoporosis, UTIs, and some that are male-specific. And again, everything is concordant across the 2 data sets that we look at. So that was a starting point. That work came out in Nature Communications in 2021. As a next step, we wanted to see, can we build a predictive model to identify Alzheimer's patients earlier? So here we integrated the clinical data with a better phenotyped cohort from the Memory and Aging Center at UCSF. And we wanted to build longitudinal predictive models, 1 day, 1 year, 3 years, 5 years, and 7 years prior to diagnosis. We wanted to understand some of the features and their clinical importance, and then we could validate quite a few of these across the UC system. So this is what the data set looked like. We start with about 5.5 million patients in the UCSF EHR. When we match to the Memory and Aging Center and then clean up the data, we get about 750 Alzheimer's patients and a large number of controls. This is what the performance of the model looks like when we compare apples to apples. So we're not comparing an 80-year-old and a 17-year-old that walk through the door. We're matching by demographics. And we can see that the model performs at about 0.7 AUC. We can start looking at the features. A lot of these are obviously ones we've seen before as associations. So osteoporosis, dizziness, hyperlipidemia, memory loss, atrial fibrillation. These are all features that are coming up in these predictive models. We can look at how these models are different for men and women. There is a number of features that are enriched in women. There are some that are general, including hyperlipidemia. Some are male-specific, and there's a large number that are specific to females, including osteoporosis. So then we wanted to see, well, how do we make sense of that? Can we better understand the biology behind some of these associations? We ended up collaborating with Dr. Sergio Baronzini from UCSF, who's built a knowledge network called SPOKE. This is a database of databases, contains about 50 million nodes, including genes, drugs, diseases, and 100 million connections of all different types. So we took the top 25 clinical features, and by we I mean Alice, this is very royal we here, and we looked at features that were common across all time points. So these little pie charts, the ones that have all colors are common across all time points. So I'll zoom into those. And we saw that there were two, familial hyperlipidemia comes up twice, two associations that are common across all the models that we've looked at. By layering SPOKE on top of these associations, we're able to get to specific genes and pathways that might be responsible for that relationship. So as we look at hyperlipidemia, we can see that ApoE is the gene that connects Alzheimer's and hyperlipidemia. Biggest risk factor for Alzheimer's, not too surprising. When we start looking at osteoporosis, though, we see a bunch of immune genes showing up, which was quite interesting. So we wanted to dig into that further. But before, we wanted to validate these clinical associations across the UC system. So for hyperlipidemia, we can see that across the UC data, we see an association that holds up using a Cox regression analysis, and we can see that it holds up in men and women. When we look at osteoporosis, however, we can see that the relationship holds up, but when we stratify into males and females, we only see this association in women. So then we turned to another completely different type of data. This is genetics data, and we looked at some publicly available GWAS for Alzheimer's and heel bone mineral density, completely independent populations, and we did a co-localization analysis. And we saw that in women, we see this peak around MS4A6A gene, which we don't see in males. This is further verified using EQTLs. Again, you can see in females, you see the peak here, and you see nothing in males. So we don't know too much about what this gene does. It's an immune gene, so involved in several immune networks. Other genes that are involved are IL-6, TNF. But really, this demonstrates an approach, a way to take clinical data, unrelated molecular data sets, connect them using a knowledge network, and come up with a hypothesis that's worthwhile testing. And really, this is in line with a lot of the vision that Atul has had about figuring out how to put different types of data together in a creative way. So now we're incorporating clinical notes into the analysis. This is a collaboration with Pedro Pinheiro-Chagas at the Memory and Aging Center, and Alice is leading this work as a joint postdoc in our groups. We're able to pull out a number of features from clinical notes using large language model outputs, and we're able to enhance our prediction not only for Alzheimer's, but also for frontal temporal dementia. So this is still a work in progress. It's up on medRxiv. Check it out if you're interested. By the way, anybody can tweet, post, take pictures. All of this is free for everybody to use, and I'm happy to share slides. There's a number of other projects in the lab that are relevant to this. We've been looking at understanding impact of social determinants of health in Alzheimer's, exploring unsupervised machine learning, and looking at specific relationships, just like we did with osteoporosis and Alzheimer's, but focusing on autoimmunity. And that work came out in Cell Reports Medicine last year. So to summarize this part, here we were able to predict Alzheimer's, or patients who are likely to develop Alzheimer's 7 years prior using HR data. Of course, we need to further validate these models in independent clinical systems. We were able to identify clinical predictors of Alzheimer's with a special interest of osteoporosis in women, and potentially link those to a biological hypothesis that needs to be further explored. I want to highlight that Alice Tang, an incredibly talented MD PhD student who led all this work, was awarded the AMA Best Dissertation Award last year and was just selected by Forbes as 30 under 30 in science. And I think she will be on the market soon. All right, I will shift gears from the kind of predictive modeling and diagnostic work and tell you a therapeutic story. And this is really an extension of the drug repurposing work that I started as a graduate student just a few years ago. This was led by Dr. Yichao Li. She was a graduate student in my lab, and in very close collaboration with Dr. Yudong Huang, who's an experimental mouse biologist in the area of Alzheimer's, and it just came out in Cell. So what we did here is we took the idea of using transcriptional reversal or gene expression reversal that I worked together with a tool with over, 15 years ago, and we applied it to single cell data in the context of Alzheimer's. We leveraged electronic medical record data to further validate these findings, and then there were extensive experimental validation too. So we always start with the data. We started with three Alzheimer's data sets, Mathis et al., Zhao et al., and Lau et al. Here, and combined them. It was early single-cell days when we came up with the hypothesis, so there's a pretty limited sample size. Of course, there's much more data out there now. We were able to integrate these data sets and came up with cell type-specific signatures for Alzheimer's. We used pattern matching against the connectivity map, which is a database of drug expression data, looking for reversal for these drugs. And as a result, we were able to identify cell type specific reversal compounds that reverse these cell type specific signatures back to normal. So this is a network summarizing these results. We started out with 1,300 drugs. 85 of them had multi-cell, 85 had any reversal, and then 25 reversed several cell types, and that's the hypothesis that we started with here. So you can see the cell types are in red, the drugs are these ovals, and we want to focus on ones that reverse 3 or 4 cell types. So those are green, orange, I guess purple is reversing 2 or more cell types. So then this is the heat map visualizing these results. We went back to those electronic medical records, that data repository information commons that I mentioned before, and we could retrospectively query these drugs to see if patients on these drugs have a lower prevalence or happen to have a lower prevalence of Alzheimer's disease. And we saw that for 2 of them, specifically letrozole and irinotecan, they came out as significant. One targets specifically neuronal cells, and the other one targets glial cells. Both of these, so this is the pipeline, kind of the screening pipeline. We went from 1,300 to 85 that reverse, 25 that reverse more than 1 cell type, 5 that validate through EMR, and then we ended up choosing 2 of those. So both of them happen to be cancer drugs. We did not go out looking for cancer drugs, but letrozole is an aromatase inhibitor, which blocks the conversion of androgens into estrogens, and it's used in breast cancer treatment. Irinotecan is a topoisomerase inhibitor, which interferes with DNA replication, and it's used for treating solid tumors. So then we staged a mouse experiment, and this is also very royal. All the work here has been done by Yichao. She's a mouse biologist whom I trained in informatics. And she did it all, from prediction to EHR mining to all the mouse work that I'm going to talk about. So we looked at a pretty strong model of Alzheimer's, and we looked at individual treatment as well as combination treatment with the drugs. And our hypothesis was, if we target the neuronal and the glial cells, then maybe we can get to a better outcome. I'm not a mouse biologist. I'm going to do my best. So the way this is tested in a behavior setting is a Morris water maze test. The mice do not like being in the water. So if you put them in the water, they try to get out. There's a target quadrant that has a hidden platform, and it takes them time. So we measure the amount of time that it takes for the mice to get out. And we could see that at both 24 hours and 72 hours, if we look at the combination, they get out faster. Yachao also measured a number of various pathological markers, looking at hippocampal volume, amyloid beta staining, p-tau, looked at microglia, astrocyte, as well as neuronal staining. There are beautiful pictures for each one, but this is really the summary table. If you look at each treatment alone, some of these markers are significant. If you look at the combination, almost all of them are significant in terms of diminishing the disease effect and pathology. As the final step to this analysis, we generated single-cell data of untreated animals versus animals treated with the combination therapy. And we could see that there is significant effects in specific cell abundance. There are specific pathways that get reversed by treatment. And this is a comparison of the original disease signature versus the reversal in the animals across the different cell types that we predicted. So it kind of takes it all back in a circular manner. We start with single cell data as prediction. We use clinical data to screen the drugs, validate them in an animal model, and bring it back to the molecular signals. So in summary, this demonstrates an extension of the drug repurposing work that we started with a tool a number of years ago. Here we expand to single cell data sets to specifically look at combination therapy for Alzheimer's. We validate this retrospectively or screened them with electronic medical records, and then carried out extensive validation demonstrating attenuation of both memory deficit as well as pathological measures for Alzheimer's. And really, this study demonstrates an incredible collaboration between computational scientists and experimentalists, and really a way that allows us to ask meaningful questions and make sure that the work that we do is potentially translatable. So we're in process of fundraising for clinical trials, as well as applying this type of approach to other complex diseases such as endometriosis. There's also been incredible single-cell data sets generated on the drug level, the Tahoe 1 million data set, and there's a lot of opportunity there. So we're actively working on incorporating those into the next iteration of the pipeline. So I hope I was able to tell you a bit about the type of work that I do in my lab, both in the diagnostic and therapeutic space, and it's an incredible opportunity, together with the data that we have, the methods, to bring all of this together and really achieve AI-driven discovery at every level of biomedicine, from DNA, molecular models, cellular models, tissue, physiological, to using population and clinical data. And Baker Institute has been an amazing place to be. We have over 90 faculty working on different types of computational projects and bringing all of this together, and I'm very fortunate to be part of it. With that, I want to thank my lab and everybody doing the work. Alice and Yachao were the two graduate students that led these projects, but it was in collaboration with so many others. A lot of the infrastructure that was allowed, especially the clinical data analysis, was developed by Atul in the first 10 years of the Baker Institute, and of course all of our funding sources. This is the lab, and before I end, I would like to read a collaborative effort. This was in collaboration with ChatGPT, and it's a poem dedicated to Atul. I was on the fence of whether to do this or not, but I knew he would have loved it. He was a technologist. He loved all the cutting-edge gadgets and things. So I'll ask for a minute of your time, and I will end with that. In quiet June, the world stood still. A brighter light no time could chill. A fighter's heart, a brilliant flame, now echoes through the halls of fame. He dreamed in data, saw in code a path where hope and healing flowed. From Stanford's gates to UCSF halls, he built an institute that calls the best to dare, the brave to try, to reach beyond, to ask the why. He shaped a field from spark to star, made health and equity stretch far. He taught us how to lead with grace, to think with fire, yet hold our pace, to mentor, guide, and build a team, and chase the edge of every dream. Awards adorned his storied name, but kindness was his truest claim. He lifted voices, cleared the way for all who sought a brighter day. A husband strong, a father proud, his love unwavering, clear and loud, his laughter light, his vision deep, a legacy the world will keep. Though cancer came, he would not yield. He battled fiercely, heart as shield. And even now his work's not done, his vision marches with the sun. So here we stand, though hearts may ache, with vows anew that we will make, to shape, to build, to seek the truth, to honor Atul's boundless youth. The world has lost a rare, bright mind, but in his wake we each will find a call to lead, a call to give. His legacy will grow and live. We all have a lot of work to do. Thank you.